Heartbeat in a dish, then the lab burned it. Here's the thing. In 1967, Paris was alive with jazz, protests, and a secret tucked inside Hôpital Saint-Louis. A team in Cellule Nine had done what no one thought possible. They grew a human heart in a petri dish. No stem cells, no bioreactors, just glass, a nutrient broth, and silence. It beat for 17 days. But wait, this isn't a story about a breakthrough. It's about what happened when that heart met a rat's bloodstream. And why, after one final test, the lead scientist ordered every sample, every note, every photograph incinerated. No explanation, no publication, just ashes. Let's start with the man at the center. Dr. Vasseur, a cardiologist who believed the heart wasn't just muscle, it was memory. He'd seen patients reject transplanted hearts, not because of tissue mismatch, but because the organ itself carried something deeper, a rhythm, a story, and he wanted to prove it. Cellule Nine was his hidden workshop, a windowless room in the hospital's oldest wing, locked behind two steel doors. Inside, incubators hummed and petri dishes glowed under ultraviolet light. Vasseur's team worked in shifts, never speaking of it outside those walls. Not to colleagues, not to families, not even to each other. By spring of '67, they had their first success. A cluster of cells, no bigger than a grape, pulsing in a dish. It wasn't a full-sized heart, just a miniature, a model. But it beat, steady, strong. And when they applied electrical pulses, it responded, slowed down, sped up, like a real organ. Here's the kicker. They didn't use stem cells. No genetic engineering, just a nutrient solution Vasseur had developed in secret, a broth of amino acids, sugars, and something else, something he never named in his notes. Compound extracted from human blood plasma, filtered through a process he called resonance distillation. Whatever it was, it worked. For 17 days, the heart lived. Then, on the 18th morning, Vasseur walked in with a rat. Not just any rat, a lab animal from the hospital's vivisection wing. Healthy, young, its blood vessels prepped for surgery. The team watched as Vasseur connected the rat's circulatory system to the heart in the dish. A single tube, no thicker than a straw, carrying blood from the rat's body into the artificial organ. And then nothing. The heart kept beating. The rat kept breathing. No rejection, no shock, just silence. For 3 hours, the two lived in tandem, a rat and a dish-grown heart, sharing the same blood. Then Vasseur disconnected the tube. The heart didn't stop. It beat faster, erratic, like it was fighting. The rat twitched. Its breath quickened. And then, in the span of 60 seconds, both went still. The room held its breath. No one spoke. No one moved. Vasseur stood there, staring at the dish. Then he turned to his team and said 2 words. Burn it. Every sample, every note, every photograph, incinerated that night. The nutrient solution poured down the drain. The petri dishes, smashed. The rat's body, cremated. No publication, no press release, no follow-up, just silence. So what did they see? Legend says the heart didn't just beat, it remembered. That when the rat's blood entered it, the organ reacted not like tissue, but like a mind. That it pulsed in patterns matching the rat's fear, its pain, its final moments. That Vasseur saw something in those 3 hours that convinced him some doors shouldn't be opened. But here's the thing about legends. They're not always wrong. In 1972, 5 years after Cellule Nine's experiment, a researcher at the University of Lyon published a paper on bioelectric memory. He described how organs, when isolated, could retain traces of their donor's experiences. Electrical impulses that mirrored emotions. The paper was dismissed as pseudoscience. The researcher? Discredited. His funding? Cut. Then, in 2003, a team at Johns Hopkins replicated part of Vasseur's work. They grew a rat heart in a dish. It beat. They connected it to another rat's bloodstream. And when they monitored the electrical signals, they found something strange. The The heart's rhythm didn't just respond to the rat's body, it anticipated it, like it was remembering. The lead scientist called it anomalous biofeedback. The press called it a fluke. The paper was buried in a minor journal. And the team, they moved on to safer research. But the archives of Hôpital Saint-Louis still hold Cellule Nine's records, or what's left of them, boxes of charred paper, fragments of notes. A single photograph, recovered from a lab assistant's personal files, showing a petri dish with a tiny, pulsing mass. And here's the open loop. In 2017, a historian digging through those archives found a reference to Vasseur's nutrient solution, a compound called Liquide Mémoire, memory liquid. The formula was redacted, but the historian found a second document, a lab order from 1967, requesting 5 liters of human blood plasma from the hospital's blood bank. The request was signed by Vasseur, and the reason listed: resonance distillation. So what was in that blood? What did the heart remember? And why did Vasseur burn it all? We may never know. But here's what we do know. Science doesn't just suppress discoveries because they're dangerous. Sometimes it suppresses them because they're too true. The heart in Cellule Nine didn't just beat. It lived. And for 3 hours, it shared a rat's blood, its fear, its death. Maybe that's why Vasseur couldn't let it survive. Not because it was a failure, but because it was a success. Cirque Cellule Nine Paris archives the truth still buried in those boxes.